Hello, everybody, and welcome to LBCA's Lobular Breast Cancer Chat Series. My name is Mason Mitchell-Daniels, and I am the Chief Operating Officer for the Lobular Breast Cancer Alliance, and I'm thrilled to welcome you and our panelists here today for a discussion on an exciting new research proposal that was recently awarded and jointly funded by LBCA and the Society of Nuclear Medicine and Molecular Imaging, also known as SNMMI. This is the second ILC specific research grant that LBCA has funded in partnership with SNMMI and with a focus on imaging and ILC. So first, let me walk you through our agenda, and then I will introduce our panelists. The format of today's Lobular Breast Cancer Chat will be a question and answer presentation with our three panelists. So now let's move to introducing our panelists and then our discussion. So here are our panelists. First, we have Dr. Ulaner, who is Chair of Molecular Imaging and Therapy at Hoag Family Cancer Institute and the James and Pamela Muzzy Endowed Chair of Molecular Imaging and Therapy, Professor of Radiology and Translational Genomics at the University of Southern California. He is dual board certified in radiology and nuclear medicine and focuses on FDG PET, CT, and novel radiotracer development for patients with breast cancer. Dr. Ulaner also serves as an active member of LBCA's Scientific Advisory Board. We also have Dr. Randy Yeh, who is a diagnostic radiologist and nuclear medicine physician who specializes in molecular imaging and therapy for cancer patients. He is an associate professor and chief of nuclear medicine in the Department of Diagnostic, Molecular, Interventional Radiology at Mount Sinai Health System in New York. Prior to joining Mount Sinai in 2025, he practiced nuclear medicine at Memorial Sloan Kettering Cancer Center and Columbia University Medical Center. Dr. Yeh's scientific interests are centered at the intersection of cancer biology and novel imaging to better target cancer mechanisms and improve the care of cancer patients. Specific to lobular breast cancer, Dr. Yeh served as the PI of an investigator-initiated clinical trial to evaluate FES PET compared to FDG PET for detection of tumor lesions in patients with metastatic lobular breast cancer prior to his departure from MSK. His experience with this trial, meeting with patients with lobular breast cancer, and discussions with Lobular Breast Cancer Alliance patient advocate Gitte Joergensen form the basis of his recent winning proposal titled Improving Detection and Outcomes in Patients with Invasive Lobular Breast Cancer Through Novel FAPI PET Imaging. And last but not least, Dr. Gitte Joergensen. Gitte has been engaged with LBCA since 2020 as a volunteer research advocate. She has participated in highly respected advocate training programs such as Project Lead, the AACR Scientist Survivor Program, and the Alamo Advocate Scholar Program. As an ambassador for the Patient-Centered Research Outcomes Initiative, she provides a patient voice and perspective in broad contexts. Gitte is motivated by her personal experience as a cancer survivor and her desire to make a positive impact on the lives of others affected by this disease. So let's start with Dr. Ulaner. Can you please tell us a little bit about your involvement with SNMMI, with LBCA, and about the Mars Shot program?
Absolutely. Thank you so much for having me here, Mason. Please feel free to call me Gary. And it's a pleasure to be working with Randy and Gitte. I have been involved both with the Lobular Breast Cancer Alliance and the Society of Nuclear Medicine and Molecular Imaging. So I guess I'm a nice segue between the two portions of this discussion. We're all very familiar with the Lobular Breast Cancer Alliance, focusing on research, education, and advocacy for patients with lobular breast cancer. The Society of Nuclear Medicine and Molecular Imaging is the world's largest society for the evaluation of imaging agents that depend upon small amounts of radioactivity, as well as therapy agents that involve large amounts of radioactivity. These agents target cancer by finding a specific molecule, which is often specific to each individual type of cancer cell, finding a key that binds to that target, and then you link that key to something that's radioactive. And if it's a small amount of radioactivity, you can administer that agent into the bloodstream. It circulates around the body, finds the target on the cancer cell, and then emits that small amount of radiation that doesn't do anything to the body or to the cancer, but allows us to see where the cancer is, because what's called a PET scanner, positron emission tomography scanner, can detect the radiation. Once we know that a target is present on a cancer cell, then we can swap out that agent that emits a small amount of radioactivity and put on something that emits a lot of radioactivity, enough to, once that it binds to the cancer cell, it can attempt to kill the cancer cell. So the Society of Nuclear Medicine and Molecular Imaging works on the development of these agents that are both for imaging and treatment of cancer and other diseases through this mechanism. Now, the Mars shot is something that was started by the Society of Nuclear Medicine and Molecular Imaging to help philanthropically fund novel research projects that have a high potential for impact in patient care once they develop into later phase trials. Dr. Gitte is the recent recipient of one of these Mars shot grants, and he'll describe his grant using the novel target called FAP, and the imaging agent that targets that FAP molecule through that mechanism that we just discussed.
Great, thank you. That is very useful. So can you tell us in your words why you think these imaging methods can be useful specifically for individuals with lobular breast cancer?
Oh, it has already been proven. So there is another target called estrogen receptor, and another agent that binds to that target. So if the target is estrogen receptor, we have something that binds to the estrogen receptor called fluoroestradiol. And that is an estrogen which has been tagged with something that emits a small amount of radiation. So this molecule, fluoroestradiol, or what we call FES, has already been shown to have value in patients with lobular breast cancer, being able to, through the specific binding to the estrogen receptor on the lobular breast cancer, detect the lobular breast cancer in patients who are were thought to have locally advanced disease, and we can find unfortunate, unexpected distant metastases that change the way the patient should and will be treated. And also it is used when there is a suspicion, when there's a recurrence of disease, and the FES can identify patients in which the lobular breast cancer has recurred, and the location of that recurrence, so that patients can get the best treatment. So there's already a model where molecular imaging and therapy has worked in patients with lobular breast cancer. And therefore, I think there's really great excitement that future targeting of these specific molecules on lobular breast cancer will lead to new and better imaging and treatment modalities.
Great, thank you. And lastly, as more research is conducted on new imaging techniques that show promise, what's the process to get these types of tests, scans, and protocols out into the world and accessible to all patients? To patients, it might seem like this process takes quite a while, but can you please explain what types of studies need to happen before they get approved and widely adopted?
Certainly. Well, it is different in different countries. In the United States, new medicines, new drugs are regulated by the FDA, the Food and Drug Administration of the United States. In order to receive a medication, the medication either has to be approved by the FDA or administered on a clinical trial that the FDA has approved. So it's either once it's initially done on trials that the FDA approves, and then you can apply for the FDA to say, yes, this agent can be used more generally by people by submitting something called a new drug application or an NDA. So the process begins with the identification of a target, then the design of a molecule that binds to that target. So this takes a lot of biologists. And then you have to take the molecule that binds to the target and radiolabel it with something that emits radiation. That takes our chemists. And then we need to be able to detect the emissions of radiation in a scanner called a PET scanner, and that takes our physicists. And then once we have all the work of our biologists, chemists, and physicists that identify a new molecule, a new target, and a new molecule that can bind to that target, and we label that molecule with radiation, we can then move into what are called phase 1 clinical trials, which are then done by our physicians and medical teams. The phase 1 trials demonstrate safety and what we call dosimetry. That shows us how much radiation is where in the body, and determine the optimal imaging parameters for using this new molecule. If the agent appears safe in phase 1 trials, then it moves to phase 2 trials. Phase 2 trials are meant to demonstrate potential. They're medium-sized trials. Phase 1s are small, phase 2s are kind of medium, to show potential that the new molecule has benefit for patients. And if a potential benefit can be demonstrated in a phase 2 trial, then it moves on to a phase 3 trial, which tend to be large trials, where often the intervention is randomized, where a patient will receive one type of intervention or another type of intervention, and one of those interventions is your new molecule, and what the other intervention is a current standard of care. And what you're looking for is to show that your new molecule works at least as well as, and hopefully better than your prior standard of care. Those phase 3 trials can take quite a long time, a number of years, and there's no guarantee that these phase 1, 2, or 3 trials will show success. And if you don't show success at each stage, then you're not going to reach the point where you can apply for that new drug application or NDA in order to get approved generally throughout the entire United States. So it is a long process: preclinical development of an agent, phase 1, phase 2, phase 3 clinical trials, submission to the FDA to get approval for it to be used generally in the United States. But even though it seems very long, it's incredibly important, because this entire process allows us to make sure that patients get the best benefits from these new medicines and minimize the risks that are associated with them. So if you want a quality outcome, you need to invest significant time and effort.
Excellent. Well, thank you. Thanks for that explanation. Appreciate it. So why don't we move on to a few questions for Dr. Ye. I'd like to first say congratulations on this recent grant award. And I'd like to ask you, can you tell our audience a bit more about your proposal, the title being Improved Detection and Outcomes in Patients with Metastatic Invasive Lobular Breast Cancer through Novel FAPI PET Imaging? And what are the main research aims of this project?
Thank you very much, Mason. And I want to thank Society of Nuclear Medicine and LBCA for this wonderful opportunity to conduct this research. I prepared a few slides. Perhaps I could share that to illustrate the points of the study, the major points of the study. So thank you everyone for joining. In short, we all know that lobular breast cancer is much harder to detect on any imaging modality compared to ductal. I think everyone here can understand that and know that from both a personal and professional standpoint. Whether it's CT, MRI, ultrasound, PET, lobular breast cancer is notoriously more difficult to detect. And we know that because instead of growing as large balls or masses, lobular breast cancer tends to form lines. And as a result, lines and growing along planes is harder to detect on most imaging modalities. So it creates a lot of frustration for both patients and the treating physicians because we don't know accurately where the disease has gone, and you can't really treat what you can't see. So our goal really is to improve imaging so we can see, detect, and then we can go after and treat. Because without visualization, it's very difficult to treat. And scientifically, what we're doing here is based on multiple prior studies that have formed the groundwork for this, including some of Dr. Ulaner's studies at MSK. And I'll speak first to the imaging studies very quickly. We know that FDG is not as good for lobular as it is for ductal. And that's because lobular breast cancer doesn't take up the glucose tracer, FDG, which is basically a sugar-based tracer, as well as ductal does. And that's because it tends to have less glucose transporters. And as a result, we don't detect it well. And when you do a traditional PET-CT, which is FDG, oftentimes you may see things, but they're very faint, or you'll see nothing, even though there is disease there that has spread, or metastatic disease that has spread. So that's one basis. Here we're looking at microscopic slides, and you'll see IDC in one panel and ILC in another panel. And this is H&E, which is telling you where the cancer cells are and not, but most importantly, FOP. So you see that in blue here in the IDC slide, there's not as much brown, right? The FOP staining is brown here in ILC. So lobular breast cancers have a lot of FAP, which is fibroblast activation protein, and much more than IDC. And our PET tracer called FOPI is an inhibitor to FAP and will hone in on those brown areas on the slide. So we thought that, this was work done in the pathology realm, but as Gary Ulaner alluded to, we want to look for molecular targets that are in cancer. So we thought, well, this target is highly expressed in lobular breast cancer, and actually more so than ductal. So perhaps using this PET imaging agent to target this target will allow for better imaging. And as we all know, lobular breast cancer in large breast cancer trials typically only form a small subset. So for example, this has been studied in Turkey, actually. So you can see the study out of Turkey. They did gallium-68 FOPI PET-CT and then compared it to FDG. And you can see clearly this patient has skeletal bone metastases everywhere. And this is these dark areas on the FOPI scan are outlining the bones. Here's the spine, here's the pelvis, ribs, humerus, or your arms and your legs here. Whereas the FDG doesn't show much, right? And you can see the drastic difference in image quality and detection between the two scans. However, of the 48 patients that were on this research study, only 5 had lobular breast cancer, right? So what we want to do is do a focused study in patients with lobular. And we know that this has been an underrepresented area in research and something that we really need to focus on. So our study, in short, is going to involve 15 patients, all with metastatic lobular breast cancer. And they're going to enroll in the trial, and they're going to get FDG PET first. And within 1 week or so, they will also get the FOPI PET right after. And really, our goal is two goals. And you can see there AIM-1 and AIM-2. One is lesion detection. So does FOPI PET show more lesions than FDG? And then the other is looking at SUV max, which is how hot or intense the lesions are. And we want to see if they show up better or clearer than FDG. And that's what we're really looking at here. However, another key aspect is really in patients with metastatic disease, if FDG shows 10 lesions and FOPI shows 20, it may not make a difference in treatment per se. So what we want to do in AIM-2 is really evaluate from both the patient and physician standpoint, did it improve their understanding of their disease and did it help? So one is the imaging component, the other is patient facing, did it improve their understanding? And then also equally importantly, did the medical oncologist have a better appreciation of where the disease has spread compared to FDG? So those two are our aims. We're going to evaluate F18 FOPI 74, which is a compound made by Sophie, and compare it to traditional PET, which is FDG here, in 15 patients with metastatic lobular breast cancer. And the second goal is really, what is the clinical value in both the patient, for the patient, and for the physicians in understanding their disease extent?
Great, thank you. So I think you touched on this last piece in research AIM-2, but I just want to ask you, I know the project has not yet begun, but can you tell us how this research will inform patient care for those facing metastatic ILC, and maybe not with this research project, but potentially in the future?
Yes, as Gary alluded to, frustratingly for all of us, there is a long process for these PET tracers, even though they show good promise, to get finally out to the clinic and to get reimbursed and done in clinical standard of care. But yes, the long-term goal is really to show first that FOPI can detect more lesions than FDG. And then also, does that translate to improved care or changes in decision-making? For example, someone who we thought did not have spread to the bones, suddenly on FOPI PET, we see it and it's very clear cut. And obviously that changes the management of the patient. And I think really what the issue or the difficulty in lobular breast cancer is that it's difficult to image. And because we can't see it, it can be frustrating for both patients and for the medical oncologist to treat. And then we can often know that, for example, some patients will have rising tumor markers and they'll get a multitude of scans that will show nothing or show very minimal things that we're not sure if that's cancer or not. And really the goal is we can treat only what we can see. And if we can see lobular breast cancer better, then that will lead to better treatment outcomes down the line.
Great. Thanks. So we have learned a bit from your presentation and also from Gary's discussion about other nuclear medicine tests like the FES PET. Can you talk a little bit more about what the difference is between what you're studying and the FES PET, just again for our listeners? What are the differences for the patient? Are there risks with these types of tests? Just a little bit more about the agent and.
Yes, absolutely. So I'll start with the conventional PET, or what we typically refer to as PET, which is FDG. And that's essentially radioactive sugar. So cancer cells tend to need more sugar than normal cells because they're trying to grow very quickly. So that's the traditional PET that we call FDG. Then there's FES, which is fluoroestradiol, and that targets estrogen receptors. So that's specific to cancer cells that are expressing estrogen receptors. And as we all know, in breast cancer, when a specimen is taken, it's stained under a microscope. It's stained and then looked at under, we do a variety of tests to see if it's expressing estrogen receptor, progesterone receptor, and HER2. And those are the key receptors in breast cancer. And this imaging agent is using the fact that many breast cancers express estrogen. And in fact, lobular breast cancer overwhelmingly expresses estrogen receptor. About 90 to 95% of lobular breast cancer tumor cells are ER positive or estrogen receptor positive. So FES has shown significant promise, and it's great because it's currently FDA approved. It's in the NCCN guidelines, and SNMMI appropriateness criteria recommends it for lobular breast cancer. And I think that it is our best agent for lobular in the current clinical setting compared to FDG and compared to standard of care imaging. There are some limitations with FES. I think the major one is that the liver has a lot of uptake because estrogen is metabolized by the liver. So then as a result, the FES has a lot of uptake in the liver. And when it has a lot of background uptake, we can't see the tumor lesions as well. The other area I think that causes some limitations is the bowel or peritoneum or the stomach, the abdominal cavity. And the reason why that's important is lobular has a tendency to spread in areas that other cancers are more rare for other cancers. For example, the peritoneum, the ureters, the ovaries, and endometrium. And as a result, if you have a lot of bowel uptake, the peritoneum is right next to the bowel, so it can obscure detection of peritoneal lesions. So I think, but again, I think that FES is currently one of our best technologies for lobular breast cancer, and it's already FDA approved, it's safe, we know how well it works. And there are ongoing studies, including one going on at MSK, where we're prospectively testing FES versus FDG to show that. And Dr. Ulaner has done work previously demonstrating that quite clearly in lobular breast cancer patients as well. So moving on to FEP PET, again, so FEP is fibroblast activation protein. And what happens is you have your cancer cells, and next to it are what's called cancer-associated fibroblasts. And they are, think of it like they are the framework, or they form the nutrients that allow the cancer cells to grow. They're part of our body, but the cancer cells reprogram them to help them. And what we're targeting is both the tumor cells that express FEP, but also the fibroblasts around it. And the thing about invasive lobular, as we showed on the microscope slides there, is there are abundant cancer-associated fibroblasts compared to ductal even, and the staining of FEP protein is very high. So we anticipate that FEP will work better and show the cancer cells very robustly. The other thing about FEP PET, or FEP PET, sorry, is that the background, the normal organs are relatively suppressed. For example, the liver, which is a problem in FES, the bone, which is relatively low. So the areas that we want to look at don't have high uptake. So we anticipate that this will allow the tumor lesions to be visualized better.
Great. Thank you for that explanation. So we understand you partnered with Gitte as a research advocate on the development of this grant proposal. Can you tell us a little bit about this working relationship and anything specific you learned from her throughout the process?
Yeah, so I met her about, I would say 3 to 4 years ago now. We had been discussing this grant and the ideas, and it was very helpful to talk to her because she gave me her experience from the patient side of what it is like to have lobular breast cancer, the treatments that she went through, the treatments that she's currently on, and her whole cancer journey. And then she told me more about what LBCA does, and then also what she does as an advocate for LBCA. So it really opened my eyes to, in thinking, how can we help from the imaging side, patients with lobular breast cancer. It also made our grant much more patient-facing. I think it made me rethink about, especially the second aim about, the questionnaire to the patients, like, was a scan helpful for you, right? Because that is important. That's something that I don't think we really study, and that's not, what's typically done in the medical literature, like how did it impact the patient, like the scan itself, right? So I think her input on both, she's a PhD, so she understands the science, which is really, it was perfect. She understood the science, she understood the patient journey, and then she also helped me better understand how we can improve the study to make it more useful for patients. So I think that combination was very nice. And we had nice discussions about other things, such as restaurants in Greenpoint, Brooklyn, where I live, and other things. So it was nice to get to know her from a personal level as well.
Great, thank you. Well, thanks so much for sharing about this project. We'll come back to you in the audience Q&A. But let me turn to Gitte for a few minutes. So Gitte, I believe you've worked with other researchers before on grant proposals. Can you tell me a little bit about what you've learned along the way in working with a researcher in general?
Sure. Yeah. So I've worked on several researchers on different grant proposals, including some of the previous collaborations with Randy. And in terms of this proposal, I just want to say again, like Randy was a great collaborator. We communicated frequently and not just about the research. So I think, to just spend a little bit of time getting to know each other and form a connection, it makes a big difference when you're like having to work together, you're building trust, and you're, really coming up with, this big, quite complex project. And one of the biggest lessons through working with any researchers for me is clear and early communication is essential. So some researchers don't always have experience with working with advocates, and they can be a little bit nervous too, It's not just us who get nervous and think, oh, we don't know what we're doing. So I think it's like really important to have upfront conversations about what you're going to bring to the table as an advocate, what you can bring. So whether it's going to be like writing the lay summaries, writing letters of support, or just providing patient feedback in general. Another thing I like to do is also just kind of let people know about my experience as a patient reviewer through ASCO and the DoD and Corey and places like that, where I have a bit of an inside view. So as a patient reviewer, we only get to look at the lay abstract mainly and the impact statements. Those are the big things. And so I think those are really worth putting in extra effort in, because that's what we see and that's what we spend a
How do
lot
I
of
turn that
time.
off?
And you can
Oops.
kind of tell if
Oh,
people didn't put an
my face
effort
on
in
here.
with the lay abstract, and it kind of bugs you if you read it and you're like, I don't know what this means, and it's written for me and I don't get it. So yeah, and then another thing is sometimes, again, researchers want to try and make it easy for you because, they don't know how much time you have and this and that, and maybe they've not worked with advocates. So they might do like most of your work, or get someone to do it, like, oh, we'll write the letter, you just have to like sign it, or, we'll do this and that. That I would, from the start, explain that you want to be included as an advocate, and we have this lived experience, and so our contributions um, are important, and we do want to contribute. That's why we're here. And that also means being able to talk about compensation or credit, such as, being included in the project or in the budget, or potentially co-present at conferences or co-author publications. So we're all kind of our own experts in different things, and that absolutely also includes the patient advocates.
Great, thanks. So I think you talked a little bit about what worked well. It sounds like you had early communication, good relationship established to make the actual grant project work. Do you have other words of wisdom that you might give to other research advocates who are interested in partnering with a researcher on a grant proposal?
Yeah,
You
so
shared a
I think,
lot already.
yeah, I mean, again, like open communication, mutual empathy, and I think for me and Randy, a genuine effort to understand each other's perspectives, so that, we're working together with a shared purpose, and we might be different, but we're also bringing like these different forms of expertise to the table. And as Randy mentioned, it's not always like clear cut, as in, he's the researcher, I'm the advocate. I have a background in psychology research, and also Randy has personal experience with cancer. So you're adding, an extra layer of experience and emotional insight to this work. And I think when you start to sort of come together on that, you can form something really meaningful. And it's not just collaborating in a formal sense, but really connecting as someone who care about the issue that we're working on. So I think, again, that's really important. Openness, talk honestly, not just about study design, but about including the patient experience. What are the challenges of, some of the grant proposals and how we can make the project more patient-centered. That's something I always try and do. And I think those conversations have been really fruitful for Randy and I in our collaboration.
Thank you so much for sharing your experience and your words of wisdom. And so that concludes our formal question and answer presentation portion for this chat. But I want to thank you all.
Thank you, Mason.
Thank you very much.